So without further ado, I would like to present to you my topic, which is the connection between methane concentrations and microbial community structures in Boston groundwater wells. So when most people talk about global warming, the main gas that they refer to contributing to this effect is carbon dioxide. But this may be because other greenhouse gases like methane are 200 times less concentrated in the atmosphere than CO2. Despite this fact, 34 molecules of carbon dioxide are equal to 1 molecule of methane, making methane an incredibly powerful greenhouse gas. And to make matters worse, it's increasing in the atmosphere. So where does this methane come from? Well, in nature, well, first let me back up a bit. Most people cite industry and agriculture as the main sources of methane in the form of waste, but there's also many natural sources of methane, such as manure, and then cow and insect guts. And for these latter two sources, the main basis of methane production comes from microbes called methanogens, which I'll elaborate on momentarily. So to put this in perspective, microbes, as we know, are incredibly small organisms, but they still contribute 69% or nearly 3/4 of the natural methane emissions each year. And they're produced by organisms called methanogens, which fall under the larger kingdom of species of organisms called archaea, and they're consumed by methanotrophs, which can be archaea or bacteria. So where can we see an example of methane causing a problem? Well, right here in Boston in 2013, BU professor Nathan Phillips measured methane levels at various spots above Boston roadways, and he produced this map down here in the lower left corner, where each yellow spike represents a methane concentration, where the concentration of methane was significantly greater than the atmospheric levels of 1.85 parts per million. And some of these yellow spikes, there's over 300 of them, and they could reach up to 20 times the atmospheric level of methane. So this is obviously a big problem. A lot of these are from the old leaky pipeline system in Boston, and many of these concentrations are reflected in the groundwater wells in Boston, which catch all the runoff water of the city. For my project, these wells will actually serve as a great model of natural methane emissions because the low oxygen, low salinity environments in these wells are ideal for the growth of methanogens. So there's a possible link here that I'd like to investigate. So now that we have this model system, I first want to know how methane concentrations and other environmental factors vary across the wells. So I went to 9 different well sites in Boston that had different fills, which are outlined here. So these wells are going to inherently have different levels of methane based on gas leaks or no gas leaks. But I also predict that factors such as fill type, dissolved oxygen, and salinity will impact the microbial community, which in turn will impact the methane emissions in these wells. And I believe that this methane will also impact the microbial communities, which brings me to my second question. How do microbial communities vary in relation to methane concentrations? So I predict that in wells without gas leaks, we'll see a relatively equal number of methanotrophs and methanogens. And then in wells with gas leaks, we'll see relatively even numbers of methanogens, but much more methanotrophs because they like to eat methane. So if there's more methane, they have more food to eat and they're going to want to live there. So as I mentioned, I went to 9 different sites. I collected gas and water samples, and I calculated the amount of methane in both the water and the air within the wells. I also collected DNA samples and extracted, purified, and sequenced these samples for bacteria and archaea. I then assigned species using a software called R, which I also use for all of my statistical analysis. So now I want to answer my first question. I'm looking at the spread of methane concentrations in gas on the top and water on the bottom. And I used the gas concentrations to remove well sites that had gas leaks, which I considered to be sites that had methane concentrations greater than 25,000 parts per million based on previous literature, denoted by the dotted line. I then removed the corresponding sites from the water concentrations, which are denoted by the purple marks below. This left me with my biogenic, what I assume to be natural or biogenic emissions coming from the microbes. And these are the points that I use for the remainder of my statistical analyses. And as you can see, there's no significant difference in the methane concentrations across fill types here. So now I'm looking at how other environmental factors are impacting the methane concentrations in the wells. So when we look at salinity, or the salt content of the wells, as salinity increases, so does the methane concentration. And this is interesting because I expected to see the opposite if these are coming from methanogens, because they typically like to thrive in low salinity. So I would have expected to see higher methane at the lower salt concentrations. So this is an interesting find. Then I also performed a correlation analysis and found that salinity increased across the 4 well types we see in the bottom right corner. I then compared dissolved oxygen to methane and found that as dissolved oxygen increased, methane decreased. This is what I expected to see because methanogens thrive best in low oxygen settings. And we also saw a decrease in dissolved oxygen across these 4 fill types in the lower right corner. They were correlated. For this slide, I'm now looking at how fill type affects the diversity of the microbial communities. So on my y-axis to measure diversity, I'm using a Shannon diversity index. All you really need to know is that the higher this number is, the more diverse the community is. And on the left, we have archaea, the right, we have bacteria, and we see that fill type is significantly impacting diversity for both types of microbes. And because we know from the previous slides that dissolved oxygen and salinity are correlated with fill type, these 2 factors also significantly impacted the diversity of the communities. And now to answer my second question, I'm looking at the specific makeup of the communities for archaea and bacteria in wells that had leaks and wells that did not have leaks, so more moderate, lower methane concentrations. So for archaea, we see that there's actually a very high relative abundance of methanogens in the wells that had leaks, and a really low relative abundance of methanogens in wells without leaks. So this is kind of not what I expected to see. And this has interesting implications that I'll get into in a moment. And then for bacteria, we see, as I predicted, there's a much higher relative abundance of methanotrophs in the wells that had leaks than in the wells that did not have leaks, because in the wells with leaks, there's more methane, methanotrophs have more food to eat. So now to get to the conclusions and implications of this data. So salinity and methane increased with one another, and other environmental trends, we saw that dissolved oxygen increased as methane decreased, and vice versa. We also saw that salinity increased across these 4 fill types and dissolved oxygen decreased. Now, just as a reminder, I predicted that low dissolved oxygen and low salinity would lead to high levels of methanogens. But an interesting find was that in low dissolved oxygen, high salinity environments, we saw our highest archaeal diversity and our highest methane concentrations. And in our low salinity, high dissolved oxygen, we saw lowest archaeal diversity and our lowest observed methane. So it's safe to assume that most of our methanogens were found in this low dissolved oxygen, high salinity environment, which is not what I expected to see. It implies that salinity is not impacting the methanogenic communities as much as I expected. Additionally, because of these unexpected results, it's likely that other factors such as fill type may play a larger role in determining the community structure inside these wells. This is also implied by the fact that we saw a significant change in the community diversity with fill type. So now going back to my second question, just a reminder, for no leak communities, I predicted that there would be moderate levels of methanogens and methanotrophs, and gas leaks would have more methanotrophs because more food to eat. But what we actually saw was that in wells with no gas leaks, we saw less methanogens than predicted, and with gas leaks, we saw much more methanogens than we expected to see. So this is interesting because it could imply that there's more methane coming from the methanogens in the wells that had higher methane concentrations than we expected. So they could be contributing a lot more to the methane levels than previously thought. So there's a couple directions that I want to take this. I actually have isotopic samples that I've sent off. I'm just waiting for the data to come back. But basically, looking at the ratio between heavy carbon or carbon-13 versus light carbon or carbon-12 in the methane that I extracted from the wells will tell me if it's coming directly from a microbe or if it's coming from a non-natural source. So looking at that will confirm once and for all where the methane is coming from in a very quantitative manner. And finally, using this data, cities like Boston can incorporate microbial methane emissions because they're more significant than we previously thought, and they'll be able to construct more accurate models of methane emissions to figure out how best to mitigate our greenhouse gas emissions in the future. So I'd like to thank a number of people and a number of organizations for funding this project, which has taken me 3 years. And thank you all for your time, and I'll open up to any questions.